There has been much debate over the use of genetically modified organisms or GMOs. Many people believe that GMOs aren't widely used, and even when they are used, some people believe that GMOs pose a potential threat to their health. This fear stems from the advertisement of organic products in the non-GMO label. The positive connotation of the word natural can lead consumers to perceive that bioengineered foods are inherently bad, as they are the opposite of natural and do not abide by the conventional standard of breeding crops.
Yes, yes, exactly. I really don't think that GMOs are natural, and that's why they should not be used. Like, I'm sure they have their fair share of benefits, but I don't want to find cancer-causing chemicals in my cereal.
While there have been negative events surrounding GMOs, scientists like myself know that GMOs are beneficial to numerous industries like agriculture and medicine. However, we do have some concerns about the impact of GMOs on the environment, specifically something called gene flow. Biocontainment systems can prevent this. These encompass a wide range of safety systems that are applied to organisms. An example is a kill switch, which are biological systems that control when an organism survives by regulating gene expression. They act as a safeguard against the negative environmental impacts that scientists fear.
This seems really promising, but kill switch is an intimidating name. Let's see what the public has to say about that.
Whoa, whoa, whoa. Hold up. Did you just say kill switch? You mean it'll kill me? Does this mean I can kill my ex-husband with this?
No, a biological kill switch can be triggered and results in the death of the host cell. It does not negatively impact a human. This trigger can be anything ranging from light to chemicals or even temperature. For example, say there was a kill switch that would activate in the presence of a specific sugar molecule. Then, when the sugar molecule is present, a signal would be sent to translate a certain region of DNA that would then start producing a cell death signal, which would kill the organism.
But if the organism is so harmful that it requires a kill switch, why should we use it in the first place?
Many bioengineered organisms serve a vital purpose that will improve our society, such as making crops more nutritious and able to grow in warmer and wetter climates. That being said, we still want to have control over these organisms. Think of it like this. We know motor oil is harmful to the environment, but it allows us to drive our cars when it's contained in the engine. Similarly, with bioengineered organisms, it might be able to cause harm if they aren't contained, but they're imperative to improve our way of living. Thankfully, kill switches are here to ensure that we have containment of these products.
I'm interested in how all of this technology came to be. Let's go back to our scientists to find out.
The study of biological systems started in the early 1800s and has resulted in incredible findings, from discovering DNA to identifying and then manipulating genes. In the 1990s, biocontainment systems used metabolic auxotrophy, which is when target cells can only grow when an outside metabolite, like an amino acid, is supplied. This would help control which cells are able to grow and divide. A cell is engineered to rely on glutamic acid, for example, and the cell will die when glutamic acid isn't constantly added. In 2006, the first clinical trial for a biocontainment mechanism to treat Crohn's disease was conducted. Then, in 2015, the first of many biocontainment systems to confine plasmids were developed, a milestone of modern synthetic biology still pursued today. One example of a modern mechanism would be gene-drive containment, which alters target genes and spreads them through a population. This new mechanism is a promising solution for disease-carrying organisms such as mosquitoes transmitting malaria. But let's take a closer look at kill switches. Biosafety systems like kill switches have been constructed to prevent the survival of bacteria outside of a laboratory to hinder horizontal and vertical gene transfer. A kill switch prevents cell survival but does not completely degrade nucleic acids. To avoid horizontal gene transfer, there are multiple mechanisms that cleave nucleic acids, which leads to self-destruction of cells. Kill switches are much more beneficial than their predecessors because their activation can be better controlled under specific conditions. For example, changes in light wavelength and temperature are powerful regulators of gene expression and are commonly used as triggers for kill switches. Currently, gene circuits are used to maintain essential gene expression, or they can induce toxic gene expression to kill a cell. Since the circuits are so complex with signaling, they're hindered by a lack of programmable environmental sensors that would activate them in a setting outside of lab. This makes them relatively safe.
Wow, there's so much to this field of science that I had no idea about. This just in, we have exclusive early access to the work of the OSU and SBU iGEM research teams, whose projects both focus on kill switches. Let's take a look at the innovative work of these two teams.
Hello, everyone, and welcome to the iGEM Breaking News. This is just in. The Ohio State iGEM team is advancing the way future scientists incorporate a form of biocontainment into their engineered organism by making it easier than ever to access biocontainment resources. They are accomplishing this by making a central database of possible kill switches, as well as making standardized procedures to further characterize and improve kill switches used by previous iGEM teams. Also, breaking news, the Stony Brook iGEM team is working on developing a kill switch for genetically modified crops that are grown in indoor farms. If any of these genetically modified crops escape their controlled environment, the kill switch would cause these plants to die when they are exposed to sunlight. This would prevent genetically modified crops from potentially becoming an invasive species in the wild or negatively altering the natural gene pool of wild plants. Thank you, guys. Back to you.
Sounds like these two research teams are doing some fantastic work with kill switches. That is all we have today for you guys on iGEM News. Until next time, have a spectacular day.